I'd like to read from Paul's letter to the Colossians, chapter 1. Giving thanks, he says in verse 12, unto the Father, which hath made us meet to be partakers of the inheritance of the saints in light, who hath delivered us from the power of darkness and hath translated us into the kingdom of his dear Son, in whom we have redemption through his blood, even the forgiveness of sins, who is the image of the invisible God, the firstborn of all creation, or every creature. For by him were all things created that are in heaven and that are in earth, visible and invisible, whether they be thrones or dominions or principalities or powers. All things were created by him and for him, and he is before all things, and by him all things consist. He is the head of the church, the body, who is the beginning, the firstborn from the dead, that in all things he might have the preeminence. For it pleased the Father that in him should all fullness dwell, and having made peace through the blood of his cross, by him to reconcile all things unto himself. By him I say, whether they be things in earth or things in heaven. And you, that were sometime alienated and enemies in your mind by wicked works, yet now hath he reconciled in the body of his flesh through death, to present you holy and unblameable and unreprovable in his sight. I'll stop the reading there. In this great section describing our Lord Jesus, we have it affirmed in no uncertain terms that he is the one who created all things, visible and invisible. We have to sometimes remember that there is a large part of creation not seen by us. There is a spirit world of principalities and powers. There are many things that are unknown to us, unperceived by the human sense, that the Lord Jesus made and is the Lord of. But of all that he made, he also became the conservator. Now, in one sense, he did that by holding all things together by his powerful word. In him all things consist. But more than that, as Michael reminded us in prayer, he became the redeemer of all that he had made. He became the reconciler of all things in heaven and on earth. And you, me, and you, he has reconciled in the body of his flesh through death. I don't spend all my time studying this sort of material, but of course I have in the last few weeks as I was thinking about these meetings. I'm not one easily moved to tears, but when I was thinking of the majesty of the creation, and then I read, the Son of God loved me and gave himself for me, it caused me to weep. We can get worship from the creation. We need to put it through the lens of the Word. But what a great Creator we have. A couple of other remarks I want to make is, first of all, if I didn't make this clear, you're not expected to regurgitate all this back to me at the end. What we're trying to create here is an atmosphere where people who are not sure of answers and are not sure they could learn them overnight would nevertheless know that there are answers. There are answers, and there are great places to go for those answers. And so it's really a confidence builder. Apologetics does a lot more for the believer than it does for the unbeliever. The only value of apologetics for an unbeliever is if that believer's mind is willing to be changed, if the Holy Spirit is working in that person, and he has a legitimate question or she's doubting something, then if you're ready and you have an apologetic that can help, you may just ease the way that they were already going, and the Spirit of God was already drawing them. If you only want to debate people, the polarities will get more and more firmly entrenched in their positions, and all it will produce is rancor. I'm not saying there's no value to debating. Again, it shows people who are listening that there are answers. But our main reason for doing this is to encourage the Lord's people. I also want to thank Brother Peter for correcting me that in the time dilation experiment on Mount Washington, it was not neutrinos, it was muons. All right? But that is a small detail, perhaps only important to a few of you.
It's not a touch screen, brother.
There we go, yes. I had to... Perfect. All right, thank you. And now if I can find my clicker, we'll move on. So the design argument I put up, I call it again by its fancy term, the teleological argument. That's from the Greek word telos, which means the end or the goal. It means that there's a reason, a goal, a purpose for things, right? So where there is a program, there must be a programmer. We know that from computer science, right? And the programming in the biological systems exists in the DNA, and a universal programmer therefore exists. Now, science is interested in finding out what that program is. People say, why do you need a God? You can take this apart and you can just appreciate that the system exists as it exists. Who cares where it came from? And you don't, need a God when you, take apart a computer to find out how it works. And we're saying that's right. A Christian scientist is going to, let's say you came across a computer and didn't really know how it operated or how it worked. And so the first thing you're going to do is tear it apart. You're going to try to figure out what the components are. You're going to try to figure out what microcircuits are and in-out gates and how silicon works and motherboards and whatever other components there are. And you're going to try to figure it out. At the end of the day, you're not going to conclude, though, that this computer did not have a designer. You're not going to look for God in the computer. You're not saying there are gremlins that are pushing the electrons up onto the screen, or that that there are little angels that are shuttling information inside. You're not looking for magic inside the machine. But what you're saying is that when you see such an obvious display of design, there needs to be a computer manufacturer and there needs to be a programmer. The thing could not possibly make sense without that. And we're going to show that the biological systems are far more complicated than any computer system. So the same logic applies. We don't look for God in the machine, we look for the God who made the machine. We don't look for a computer programmer inside the box, right? We don't expect to find that person hiding under the lid here. But we know that person exists. Okay, so in the beginning was the Word is how John says it, but I'm going to say in the beginning was the program, because the program, and again we relate to this in our computer age, is exactly the kind of thing that the Lord installed into the system to make it work. Computers, as you know, use a binary information code, either an I or an O, in, out, right, up, down, is a toggle. It's a binary system. And in doing that, they come up with sequences, and I gave an example there, 1, 1, 0, 0, and so forth. The order in which they are arranged, of course, in the storage media tells what the information is so that it can be retrieved. DNA, as we're going to see, is a long molecule as well. It's not a magnetic tape, but it contains information in linear sequence. But it doesn't just use in, out, or 1, 0. It uses 4 different letters. We call them A, T, G, and C. And so it becomes more of a quaternary compound, not a compound, but a system of different letters. And of course, you can store much more information using 4 letters than you can just using 2 numbers. Much more to say about this. If you're not familiar with it, I hope to make it clear. But DNA is not just a molecule. It's a molecule that instructs. It's a physical substance, but it contains something immaterial. That immaterial is information. That information is encoded into that amazing molecule. So we're talking about informatics here. That's the understanding of information and how it is used in systems. It always requires an intelligent source. It's always communicated in some way, in some kind of language, and it is not material. It is something rather that is imposed upon material to guide and to form material. And if that's not clear, let's just look at these 3 well-known entities, right? If this is what, let me instruct some of you younger ones. This is called a cassette, okay? And that's why we tape these sessions, right? And this is an old obsolete dinosaur now too called a CD-ROM, right? And this one is certainly not a new thing. That's a flash drive, jump drive, whatever your favorite expression is for that. Now let me ask you something. If this CD is full, or if it's empty, is there any difference in its weight? Is a CD that you've chock full of information, does it weigh any more than an empty one? No. No. All that has been done is there has been a code, a language, an order imposed on the physical structure that is coding for something that is not itself material. This is extremely important for us to understand biological evolutionary debates. because the existence of such a highly ordered program has no explanation in the random movements of molecules and in the chance occurrence of events over time. And here's a kind of a stylized picture of DNA. And here is a couple other depictions of it. You can see it is a helix, or a spiral if you wish. It has two strands. We call that the sense and the anti-sense strand. And in between are these little struts that are composed of, really, they're just bars here, but they're two different molecules, A pairing with T, G pairing with C. Those are the names of adenine, thymine, guanine, and cytosine. And here is a molecular model where we have the different atoms as little balls of color, depending on which one we're talking about. This is very much like a zipper, okay? So you unzip it, you can read the code, and then when you're done, you zip it back up, right? And you store it in its zipped form. We're going to also see that the way the body uses this, it doesn't make proteins directly off of DNA, which is the master tape, but it makes protein off of a working tape. So the first thing it will do is unzip the DNA, and then it will read the code and make a template, a working template. We call it RNA. That RNA will then move off into a machine called a ribosome. And that machine, we're going to see an animation of this in a minute, will then take amino acids that are brought to it one at a time by little shepherd molecules we call tRNA, and it's going to make the exact protein sequence that was coded by the DNA. Now, the working tape is disposable, right? But the master tape must be carefully protected. And there's a whole variety of molecules that will zip it and unzip it. They'll twist it and untwist it. They'll look for mistakes and fix them. And they'll just protect it in a very, very orderly and wonderful and amazing way. Each gene encodes information for a protein. Something was on top that you can't see in the white, but that's okay. How big is the DNA in each one of your cells? About 1.5 gigabytes of data, 500,000 pages of text. In that little cell I just flicked off. Okay, pardon me. Have to be swept up later. But every one of your cells, except for red cells in special situations, have 1.5 GB of data inside of them. Okay. What is not seen is not important. We're going to move on. Okay. Here is a set of chromosomes. So what are these chromosomes? Well, these chromosomes are the way your DNA is packaged. You see, if you took the DNA in each one of your cells and strung it all the way out, it would be 6 feet long. That has to be coiled up, and it has to be controlled in an organized way. But not only it has to keep it from tangling up like spaghetti, but you actually have to be able to go to each of those volumes, be able to pull the book off the shelf, so to speak, open it up, get the code out, use it, put the book back, and make sure that it's properly replaced where it came from, right? So this is very complicated. So humans are diploid 46. That means we have 23 chromosomes. And in this case, I actually have an abnormal situation. I have 3 chromosome 21s. That makes this the genome of a person with Down syndrome. We also know that it's a boy because there's an X and a Y, right? If it was a girl, there would be 2 X's. A little bit of trivia. But that's how these DNA molecules are stored into these volumes, we call them, or books in a library shelf called chromosomes. Now, the way this works is that you see the DNA working in its helix and it being taken into a cell and packed into these chromosomes again for storage, if you will, until either the cell needs to reproduce, in which case it lines these chromosomes up. You can see them stained here, and then miraculously pulls them apart. How does that work? Well, there's a whole system that allows the cell, after it has duplicated its chromosomes, to line them up along a line with no mistakes here. Each one is paired with its partner. And then there are microtubules that pull them apart. Works every time, okay? And then the cell will seal up in the end, and we now have a clone. We have a new cell with the identical genetic makeup of the original one. So that's pretty awesome, right? Did that happen by chance? I don't think so. All right. Here's the unzipping of DNA and a little animation for you. So you have to get at this code, which is going to be held on the sense strand, the other strand we call the anti-sense strand. And of course the machine has to know which side has the proper code. And then what we're going to do is make messenger RNA. So this DNA polymerase comes along here, and it's going to make this strand of RNA by reading the DNA code. And again, the animation is just to show that this is a dynamic system that works quickly. And there's our code coming down, which can then be used to make protein. If we move to this slide, we see some of these shepherding molecules that are needed in order to keep the DNA organized, keep its information accessible, keep it from developing mistakes or mutations that would be dangerous, and to figure out what to do. If you've ever dealt with a long hose, like an air hose, or a garden hose, how frustrating it is when it gets those twists in it? No worries. There are molecules that know how to untwist this thing. Okay, they're called topoisomerases, ligases. There are gyrases. There are different molecules that know how to deal with the twisting problem that we have with our hoses. Okay, and so we have, I'm not going to go into the details of this, just get the point. It's complicated, all right? It's very complicated. And by the way, every one of these things itself was coded as part of the DNA. So the DNA has the code to make the molecules that take care of it, that know how to replicate it, that know how to translate it, right? Transcribe it, we say, and then that's translation into protein. This is getting into some details, but this is an important thing. This is DNA replicating itself. A completely different set of enzymes now that do this. So that, for example, the cell can divide. So if you want 2 copies of this DNA, you're going to have to completely transcribe it from one end to the other. So you now have 2 chromosomes where you had one before, so that you can separate them into 2 cells. But when you're going to make protein, which is the purpose of the DNA, then you're going to have to read the code, and you're going to have to make messenger RNA, a different set of enzymes, and then these molecules here are pulling amino acids together to read the code. And here's an example of a little tRNA, it's called. It's a molecule that knows how to connect to an amino acid called methionine. And the start codon for any transcribed protein, with very few exceptions, is always going to be methionine. So that's the first one. It comes to the AUG, start codon. What's a codon? A codon is a group of 3 base pairs that stands for 1 amino acid. Interesting, the stamp of 3, again, in creation. And so when that happens, along comes this subunit here, which I guess when I push this, I guess I don't change it. It's bringing, this is the amino acid, that's the shepherd molecule. These are 3 base pairs that are going to code to those 3 that match them. U to A to U, C to G. Once it's in place, that amino acid is kept until the next one comes, and then, of course, it's released from the tRNA, and the next protein will then be released by its tRNA, and this daisy chain, this string of pearls, will continue until you have your peptide made. Bottom line, for those of you gazing at this, is it's complicated. All right. Here is the genetic code. This is the genetic code that tells you how to make protein, which is exactly what DNA does for you. Once you've made the protein, you can deal with fats, you can deal with carbohydrates, you can deal with metabolism, you can deal with all kinds of things, but you start with proteins first that know how to then, in a secondary sense, assemble every other macromolecule your body needs. There's our start codon there, which in the RNA language is AUG, standing for the amino acid methionine. There are also special stop codons and a bunch of other details, which we will not go into in our time here. So this is the depiction of a protein that's just been made from DNA, and it's all folded together. And these folds are based on electrical charges and other interactions. This might be a hemoglobin, right, that's able to carry oxygen to your cells. This might be an enzyme. This might be even a shepherd enzyme that's going to come back and control the DNA. So living things are by far the most complex objects we know of in the universe. The E. coli bacterium, that's a bacteria many people have heard of, right? You don't want it in the drinking water. You don't want it in your lakes where you're swimming, but when it is where it belongs, it's fine. It has 10 to the 12 bits of information. That is the amount of information needed to make that one-celled. This is an organism that doesn't even have a nucleus. Right? It's the simplest kind of cell. Is 10 to the 12 bits, 100 trillion pages of fine print. Now that's not just what is coded in the DNA. That is all the information needed to make the entire simple, right, Darwin? Simple organism. The thing that just is goo, that is the basis upon which evolution can play. This rules out chance. Humans have trillions of cells that are far more complicated than E. coli. The information content is beyond even the probabilistic resources of a universe given billions of years. The subject of mitochondrial Eve is a very kind of an advanced one. I'm only going to mention it because there is a recent publication in Science that if one takes the proper, and I say the proper presuppositions, you come to the conclusion that the woman who is behind all other men and women, the so-called mitochondrial Eve, was on the earth about 6,000 years ago. Amazing. Now, you're not going to read that in all the science textbooks because they won't accept that a priori because, remember, they're being deductive, right? Not inductive, deductive. So they begin with the premise that the earth is old and evolution is true, and then they try to work with this, and the fixes, the patches, the sort of explanations they come up with are very unsatisfying. You have to use radio dating methods, which we're going to talk about tomorrow, and you have to make certain assumptions about relationships to chimpanzees and other organisms to come up with an older age. Because it's an advanced topic, I'm going to leave it other than to make that single point. So we talked at the first meeting a little bit about the difference between microevolution and macroevolution. And I said, although I will use those terms, I'm not entirely thrilled with them. Because it seems that the difference between micro and macro is one of size. If it's small, it's micro, and then if it's big, it's macro. But the real distinction between true evolution and false evolution has to do with the plane in which it occurs. Is it horizontal or is it vertical? Does it work with existing genes in the gene pool, existing alleles, I used that word yesterday, or does it create new genes, new chromosomes, new genetic material, new information by chance, and add it to something that already existed? And the answer to that is that if it's on the horizontal plane, the changes could be big or they could be small over time. There could be a point where speciation occurs, where two populations have been separated on their horizontal plane, still in the same basic kind of plant or the same basic kind of animal, so that they can't interbreed anymore. That certainly can happen. That's still consistent with the biblical view of, we're going to call it microevolution. Okay, so variation, adaptation on a horizontal plane. There's abundant evidence for that. We have no issue with that. Now here's the problem. They're going to extrapolate that and say that if it happens on a horizontal plane, it's going to happen on a vertical plane. If you can work with an existing kind, you should be able to make a new kind. If you can manipulate current DNA, you should be able to manufacture a new DNA. And the problem is there's no theoretical way that could occur. I mean, they have their theories, but I'm talking about in terms of anything demonstrated in a scientific laboratory, number one. And number two, there's no examples of it in the fossil record. They say there are, but all they're showing is variation on a horizontal plane. They come up with these fabrications like the horse series and the whale series, and that's downstairs in the museum where all the school children are coming and learning. Upstairs, the scientists are saying, and I have the quotes here, it's a bunch of malarkey. This is absolutely a fabrication. It's a story imposed on the evidence. And they, in some cases, Lewontin and others, are chagrined that this is passing as the truth when they know that there is almost no reason to consider it to be true. Here's an example of adaptation, antibiotic resistance, okay? So we have the story of penicillin. You know, Jenner, some of you are old enough to perhaps remember when penicillin first came out. It was a miracle. There are people who are on death's door with pneumonia in both lungs and just about ready to slip out. The death rattles and everything were there. They were all full of, almost moribund, and they came along and gave them a vial of penicillin, and in 2 days they were better. We never see that anymore. Okay, never see that anymore. In fact, sometimes we give them penicillin and nothing happens. Why is that? Because the bacteria are resistant. Well, how did they get resistant? Did they create new genes by chance? No. For the most part, resistance means damage to current DNA, damage to current substances, or in some cases they can borrow DNA from another organism, but in that case it's still a horizontal change. So if you look at the examples on the slide, penicillins and cephalosporins and bacitracin and vancomycin inhibit cell wall synthesis. Other types of antibiotics will come in and they'll affect transcription, translation, or protein synthesis, or other important functions in the cell. And so that shows where these occur. And then here are some examples of how resistance can occur. There could be a change in the port. There could be an activating enzyme. This is different. There could be an enzyme that is able to cut the drug in half, and it increases in frequency in the gene population. That could occur outside or inside the cell. There could be an alternative enzyme pathway that gets around the block created by the antibiotic. There could be alterations in the target if they're going after DNA. There could be pumps that exist already. They didn't create them out of nothing. They already existed in the gene pool, but now the bacteria that have them are going to survive, and those who don't are going to die, and this is going to become very, very prevalent quickly. And these generations occur very, very quickly, over hours, right? And so these are mechanisms we have to deal with, and we're constantly trying to outsmart these bacteria. And in some cases we're losing, and in some cases we're just holding our ground. Again, this is a little bit advanced, but another slide, and here's another one that gives a lot of the different antibiotics, how they work, and places where antibiotic resistance develops. So, this slide says, if you can't read it, psst, kid, hey kid, want to be a superbug? Stick some of this into your genome. Even penicillin won't be able to harm you. It was a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. Okay, another example of variation is in eye color, right? Many different eye colors. This is not controlled by a simple set of genes, but in general, lighter colors like blue and green are going to be more recessive, and darker colors like brown are going to be dominant, which means, I think most of you know this, that if you are going to have a lighter colored eye, you need to have, say, at least two, let's say, keep it simple as a two-gene system, you need to have two blues. If you have a blue and a brown, you're going to be brown or intermediate. And of course, if you're two browns, you're going to be brown. So these are simple ways in which we understand change in populations moving from one side of a horizontal level to another. Evolution doesn't say where eyes come from. Right. It can say whether brown or blue, but it can't say where they come from. And again, we talked about our dogs on the first lecture, and we talked about our finches. And now we want to show you the tree of life. A lot of people naively think that the Bible teaches that God created greyhounds and dachshunds and boxers, although most people wouldn't go that far. But they think he created dogs and dingoes and jackals and foxes. And it's not always exactly clear where the kind boundaries exist. We have bounded variation in biology. That bound is a little hard to define at times. It certainly isn't bound by our definitions of species and genuses and so on. But there is a kind that God created, only one. And from that, there was a branching that occurred. So this is incorrect. Whoops, got to get my arrow going here. Where every single kind of animal was made individually, and you see some of these are short lines, which means they went out of existence. But that nothing was ever seen on the planet that was not directly made by God on the first week of creation. That's not true. I don't think that's true. I don't think that's what the Bible says. I don't think that's what horizontal adaptation teaches us. The other extreme is this one that says all life came from a single organism that spontaneously arose. Or if you're like Francis Crick and realize there's no mechanism on earth to explain the origin of life, you say it came from outer space. Is that not kicking the can down the road again? Does it not beg the question, where did life come from in outer space? But since we don't know because we can't get there and we can't access that information, we can just make assumptions. Well, it did. After all, there are living things on Earth, so of course it did. It came here on a meteorite. And it found its way into a little pond, and from there, evolution took off. That was cynical, but it's actually the truth, all right? So they say this. We say, no, this is what happened. This is what Genesis teaches. After his kind, this is what science shows. This is what the fossil record demonstrates. God creates a kind. Sometimes it stays fairly true. Other times it has extensive branching. But all of these, even though this is an up-down orientation on this field, are horizontal changes conceptually. That is, changes within the gene pool of an existing kind. You with me on that, right? So sure, adaptation occurs. And some organisms didn't make it too far. If evolution were true, you should have few organisms at the beginning and many, many more species today than existed even 1,000 years ago. Certainly than 100,000 years ago by their time reckoning. You see just the opposite. The number of species today is much smaller than before. Why? Because things have degraded. Species have gone out of existence. Things have gone extinct. And again, this is into creationist jargon. Now, there is such a thing as creation science, okay? And, of course, secular science don't have much respect for it. Some Christians don't have much respect for it either. They say, let's just stick with God's Word. And actually, I have sympathy with that. I say, yes, let's just stick with God's Word. That's clearly the beginning and ending point of the discussion for us. But there's nothing wrong with inquiring in your mind and using the principles you know to be true from Scripture to see how well it really comports with reality. And when you do that, you find that these answers fit very well. So they come up with what they call baramins. Now you have to know a little bit of Hebrew here. Bara is create. Min is kind. So these are the created kinds. Yes, creation scientists have their jargon too, like everybody else, every other technical discipline, and then they intimidate other people who don't use the same lingo, right? Don't be daunted. It just means the kinds. All right? And here's man, a monotypic kind. He begins with one race and he remains one race. Yes, there are changes in skin pigment. And at the flood, we have another node, right? Because everything before the flood of the race of Cain was lost. Noah, his three sons, and his 3 daughters-in-law come out of the ark. And from them, there is a variation in mankind, but there is only one race. A polytypic example would be the ass-like or donkey-like variety of horse kind and the horse-like variety, which may have been separate from the beginning and never have interbred. And then they, in turn, diverged. And again, you get to the flood, you go back to a single node, and then the various kinds. Here you have to read them on the side. The quagga, the zebra, the horse, the mule, which, of course, is a sterile crossover of 2 different branches of this tree. Sterile because perhaps right from the very beginning, the ass-like and horse-like kinds were different. This is all surmise, right? This is just logic, sense, reasoning, but it makes good sense. A polytypic baramin might have been a fox-like, dog-like, and hyena-like original kind, which remains very easy to distinguish, although they certainly have more in common with each other than within the other organism. So again, what happens to the dog kind? Well, we begin with the quintessential dog, which I suppose is a wolf, and then we have coyotes and dingoes, and finally the breeders come into play, and we have collies, still a noble breed, and then we have that. But it's still a dog, okay? So it's still a dog. Still a creation of God. We'll respect it. Notice the arrow is down, right? Down, 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 down. And again, look what occupies the lowest rung there. This is evolution, okay? And, of course, this is easy to find in textbooks, impossible to find in real life. We're going to talk now about irreducible complexity for the next 20-25 minutes. Here's William Paley. I'm going to go quickly through this because I do want to get through some of the later slides. Paley is famous for saying if you found a watch on the beach, you wouldn't guess it came there by chance. You would say there's got to be a watchmaker. Dawkins comes along and says, no, there's no watchmaker. You just think there is. It looks like there should be, but he's only the blind play of chance, the blind watchmaker. Well, again, we go back to straight logic. Where there is design, there must be a designer. Design exists throughout the universe, therefore a universal designer, we call him God, exists. The Bible uses this argument. Therefore, we shouldn't be afraid to use it as well. Psalm 8, I look at your heavens, the work of your fingers. I know you exist. The heavens declare the glory of God, he says later in Psalm 19. So let's go to another psalm, well-known, 139. You formed my inward parts. You knitted me together in my mother's womb. His invisible attributes, his eternal power and divine nature have been clearly seen. We've gone over some of that ground already. In the beginning, we said, was the program earlier. Now I'm going to say in the beginning was the design, and it really means the same thing. Nature is full of these locks and keys, males and females. Well, it's a bit of a delicate subject to talk about publicly. One of the biggest unexplained realities in biology is the existence of males and females. There is no good evolutionary explanation for that. In the beginning, he made them male and female. Notice the Lord Jesus is a young earth creationist. From the beginning, in the beginning, he named them male and female. From the foundation of the earth, the blood of righteous Abel. Righteous Abel, male and female, at the very beginning of creation, when the foundation was laid. Well, there are people like him, Thomas Huxley, there are people like him, Richard Dawkins, who don't see it that way. But they both admit that it certainly looks like the universe was designed. It's just that if you really have the secret information that they're, of course, trying to peddle, you would know better. We've been through that already. So we're going to talk about intelligent design. Design is too obvious to ignore. This is Apollo 11. Apollo 11 made it to the moon. How did it get to the moon? Do you suppose it might have taken millions of man-hours to design and work out that project? Do you think there were prototype rockets? Do you think there was experimentation? Do you think there were a lot of slide rules sliding back and forth in those days when they didn't have calculators, right? Of course there was. Who is able to design the whole universe if it takes mankind that much time to design a very simple system called a rocket? The answer is God. We detect design all the time. Here's an example, again, pardon the North American focus of the slide set, but that is Mount Rushmore. And for those of you who are not, whoops, I keep pushing the wrong thing here. Abraham Lincoln, Thomas Jefferson, and of course, George Washington. I'm the American here. George Washington, Thomas Jefferson, Abraham Lincoln. And that's Theodore Roosevelt, who happened to be president at the time this was carved in the limestone of Mount Rushmore in South Dakota. This is a arrowhead. You find that, you know that's not just a random rock formation. There's somebody who's made that. And if that is true, we detect that all the time. We know that every house is built by some man, according to Hebrews 3, and he who built all things is God. The properties of design that we're looking for are complexity and specification. Now, there's a computer circuit board there to show that this is something we very much relate to on a very simple plane. The object in question cannot be so simple that it could have been explained by chance. Second, the object contains order and displays patterns that require an intelligent design, an intelligent mind. I'm not going to waste my time on more Dawkins material. Let us work on, and this is of course Darwin naively thinking that the cell was no big deal. But we want to talk about this concept of irreducible complexity. And there's going to be some animations in the next few slides that show molecular machines. And of course these are mock-ups, they're not actual pictures, but they're made from electron microscopy. And so we have here the cell membrane, which is a lipid bilayer, which means it's greasy on the inside and it's water-soluble on the outside. And spanning this membrane, this would be the inside of a cell, and this is the outside, is this machine, which is being powered by the flow of protons, producing ATP, requiring changes in molecular shape, not only back and forth motions, but true rotations. Note the complete 360 spin. Each one of these rotors here is made up of subunits, every one of them coded by a gene in your DNA. And this machine is producing things that are being flipped off at the bottom. And I don't really care that you know what exactly is being depicted, just to get the idea that this is a molecular machine. No part by itself has any utility whatsoever. There's no evolutionary pressure, no selective advantage to make these expensive little subunits and to have them floating around waiting for the opportune time when all 12 of them finally are in the proper orientation to know how to self-assemble and then form this machine. We move from improbability to impossibility. We move from something that is highly unlikely, even over a long period of time, to something that could not occur no matter how much time you dump on it. Because this is not possible by evolution's own principles. It is not possible to expensively make and maintain numerous subunits which have no utility until the whole thing is together. There's another depiction of one. And again, these are supposed to be molecular machines, irreducibly complex. So let's grapple with this expression, irreducible complexity. It means that the complexity is such that the whole machine and all of its parts must be present, every part accounted for, every part working properly, or the whole thing is useless. It cannot be reduced. It cannot be reduced to something less than what it is, because it has no function unless it is everything it is all at once. We lost something there. That's all right. This is a mousetrap, and it's a good way to illustrate this. A mousetrap is made up of a board, a hammer, a holding bar, 3 staples, a spring, and a bait plate. And in this case, Swiss cheese. Now, if you were to evolve as a mousetrap, how would you do it? Well, you would have to make each of these parts separately, and they would have to have some reason for being made and reason for being maintained. Until the last one finally came into existence, and then you would have all your parts. But you still have the problem of self-assembly. How does the mousetrap know how to put itself together? Well, once you get over that obstacle, then after the number of millions of years that it took to make all these parts, you finally have a working system. And now you can catch your mice, right? Now, that is ridiculous beyond words, what I just said. There's no way there's any mechanism to account for the production of all of these very simple parts to a very simple machine. And this is child's play. This is nothing compared to what we're talking about at the molecular level. So, 19th century evolutionists thought that the cell was no big deal. We know that just as we look out with a telescope and see far more and more and more and more and more complexity, more and more majesty, more and more mind-boggling dimensions. Do you know all of us could, with the number of stars that we know exist today, each of us could own 11 trillion of them and still have some left over. Well, we've done the same thing with the microscope. The smaller and smaller and smaller we get, the more complexity, the more organization, the more wisdom, the more irreducible complexity that we see. So it's really ironic that in the age of science, the evidence for creation has multiplied and it has increased exponentially. It has not decreased, it has increased. But there is none so blind as he who will not see. Okay, there is a depiction of a bunch of things in a cell. We won't dwell on that right now. This is an actual transmission electron micrograph of a cell, showing a lot of these different components. A lot of you who had biology will recognize it as a Golgi apparatus. You'll see some endoplasmic reticulum. You'll see some mitochondria, right? And so these are all machines inside of, not machines, these are organelles inside machines. Sorry, say it again. These are all organelles, little parts inside of a cell. And each of these parts, in turn, has multiple molecular machines. An artist's depiction of the same, and again, this looks complicated, but this is all simplified to make it learnable. It takes a while to learn all that stuff, huh? Now, there's just a little depiction of biochemistry for you on a page, right? Those are biochemical pathways. And yeah, if you're a biochemist, you're supposed to learn all that stuff, or at least know where it is on the wall so you can go find it quickly. And at the beginning, at the middle, we have this sort of wheel here, the tricarboxylic acid cycle, commonly called the Krebs cycle, where energy is made in cells. And my point here is not to dwell, but to kind of move through this and show you this is complicated. There's the citric acid cycle, as it's sometimes called. There's the combustion equation. So your body is just like your car. It takes a carbohydrate, a carbon backbone, and it burns it. And when it burns it, it gets oxygen, say that again, carbon dioxide, water, and heat, right? So what comes out of your tailpipe? Carbon dioxide, water, and heat, right? That's the result of breaking down a hydrocarbon, like CH4 methane, or C6H12O6, which is glucose, sugar. The thing is, you don't have to burn. You don't have to have flames. You don't have to have flames coming out of the the machine that's making this, because this combustion is controlled. Still produces heat, which we use, and it produces usable energy. With formatting changes, which are probably a mercy at this point, a lot of what's on the slide has been lost, but that's okay. You see the depiction here of a human eye. We have the transmission of light, which comes through the clear cornea. Why are corneas clear? Because they have to let light through. Duh, right? But then, how do clear corneas arise by chance? Once the light comes through, it must continue to go through a clear series of two chambers, both of which must be transmissible for light, right? They must be clear. They must be pellucid, we would say, if you use a fancy word. But, if there's too much light, that would be bad, or too little, that would be bad. So you need to change the aperture. You need to have a way to close down the light or to increase it with the lens, right? The iris, we're not at the lens yet. The iris, right? So that's the colored part of your eye that allows a certain amount of light to come in or out automatically. Then it gets to the lens and it has to be focused. It has the parallel rays of light coming in have to be bent, and they have to be sent down to arrive in sharp focus, hopefully, on the fovea centralis, which is a piece at the back of the eye which has a great deal of a certain type of cell, a photoreceptor, we call a cone, which is able to visualize color because it has red versions and green versions and blue versions, and you put those three together just like your computer screen and you come up with a color. Then there are other cells that are more on the periphery of the eye that are very good at detecting light and dark, and they're much better for night vision. And of course, some of them, they are so sensitive that they are able to detect a single photon coming into your eye. English did show up. Okay. The eyes handle an astounding 1.5 million messages simultaneously. Now, if you think your eyes are good, you should check out the eagle's eyes. The eagle can see 8 times better than you can see. He has a 340-degree field of vision. 360 would be all the way around, right? So 340 degrees. He can see a rabbit 2 miles away. Everything that he sees is magnified and made brilliantly colored by his amazing eyes. So the thing says here, if you had an eagle eyes, you could see an ant on the sidewalk from the top of a 10-story building. I had flowers. Well, we put flower design. You've all been through that in botany. Let's remember, to get an insect to come into this plant, you have to have a yummy scent. You have to have nectar that makes it worth coming in. You have to have a color that will be attractive to that insect. And you know what? If all of that was created on day 3, and then 1 million years occurred before day 4, you got trouble. They say if all pollinating insects were killed right now, the rest of us would be dead within 4 years. So pollination had to occur at the same time that the flowers occurred, right? So that the flowers could survive, but also that the insects themselves would survive and be there to continue this great process of pollination. A couple of more molecular motors to finish off. We're nearly done. This is then we're going to talk about the bacterial flagellum. Now, to flagellate means to whip, and of course, this is the word in Latin for a whip. Now, bacteria, they're like little speedboats. They know how to drive around, and they can propel themselves with whips. This is a real photomicrograph of one with a little color enhancement, and you see this whip coming off here. Some of them have many whips, and they can coordinate these into something that's much more powerful for propelling. These propellers spin 360, and they can be instantly reversed. So if he's moving forward, I don't know if it's a he or she, they don't have gender, okay? These are little protists, okay? But they move forward, and then let's say they want to stop the forward motion. They simply reverse the motor, and they tumble. Again, you can see the same thing with those that have multiple flagellae. They know how to coordinate them, and they rotate as one until the bacterium wants to stop. Then they all reverse gears, and they tumble. This is what the bacterial flagellum looks like. And again, it makes the same point as the molecular machines I showed you earlier, except this time we're going to actually label what's what, okay? So we have an S ring and an M ring, which create a rotor. We have bushings here because of the membrane span. And we have the shaft. We have a universal joint. We have the propeller out here. And so all these things have to work together. This is an actual electron microscopy picture of this. So you know we're not making this up. And again, this is a depiction of it. And now you get to see a little animation here. Watch this guy on the right. Right here you see the rotor. Whoops. Don't worry, I know how to do this now. You see the 360-degree rotation. It's actually going that way, which allows this thing to flip and to whip. This is the flagellum coming out. We're going to see a cutaway. We now see the engine, the rotor, all of its parts working. That required engineering that's beyond the mind of any human. Again, another depiction of it. I think we'll go on. The point I want to make now is that there's a very similar projection, organelle, we might call it, or method of either motility or of moving substances along a membrane that occur in eukaryotic cells, that is, in human cells. Now, if we evolved from bacteria, we would expect that we would have that bacterial flagellum system brought over into the human cell. But no, God decided to start with an original design, completely different than the bacterial flagellum. This brings up my one point where I will mention the idea of homology. People say, well, look, a lot of animals, frogs have 5 fingers, and we have 5 fingers, so we must have come from the same source. Well, there are 2 answers to that. Number 1, we're all living in the same world. We need grasping abilities. There's no reason why a good design couldn't be used in more than one animal. But another argument is that the frog's hand and your hand develop entirely differently. You say, how so? Well, your hand starts as a mitten. And then through what is called apoptosis, programmed cell death, the cells that form what are now the divisions between your fingers die off. And you're left with 5 fingers. That's not how a frog makes his fingers. A frog starts with a fist, and the fingers grow out as projections one at a time. So it looks the same, it functions the same, but its design is entirely different. Same here. If we had come from bacteria, we would expect that our cilia would operate like bacterial cilia, and we would have borrowed them from the bacteria. Nothing could be farther from the truth. Without going into the details because of the time, we have a highly complex system of ratcheting microtubules that move our flagellae back and forth. Actually, it's usually not flagellum. We don't call it except for the sperm cells and a few other mobile cells. For the most part, these are made to be the reason. So let's say you have a respiratory epithelium here, right? So it's the lining of your lungs, and you've got something that's gone down into the lung and has caused a problem, and you need to bring it up. You need a conveyor belt to bring it up, right? So all these cells, they're all pushing things in the same direction. Pushing, until finally, in a coordinated method, they bring the substance all the way up through the trachea to the throat where it can be either spit out or swallowed. Now those little cells are just doing their little job, right? There's a master plan here because each of these cells doesn't know about its neighbors. It doesn't know why it's moving the mucus. It just knows it does that because it likes to. And it's the way it was designed, right? So the way it does that is through something that superficially resembles a bacterial whip, but in fact is a human design or a mammalian design that is entirely different. There was an animation on this, but we won't stay with it. This dyning arm becomes very taut as the power stroke occurs, and essentially this is a ratcheting mechanism where by continued motion the system goes click, 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 click over. This is a depiction of it, a bit of animation. This was my thing about homology. Last section. I'm only going to take 2 minutes on this. A lot of human engineers have realized how brilliant a designer God is. And they have decided instead of beating them, let's join them. Okay? This is called biomimicry, where you take a really cool design from nature and try to use it. Now this is the Shinkansen. 500 looks really sleek and aerodynamic. The only problem is when they emerge from tunnels, they create the equivalent of a sonic boom. Because of the difference in air density in the tunnel and the density outside, as well as the pressure in the tunnel and the differential with the pressure, as soon as the engine emerges from the tunnel, there is the equivalent of a boom. And that, of course, is a big problem when you're trying to get into noise abatement. So how do you redesign this so that there's not going to be any noise when it leaves the tunnel? You go to the Kingfisher. Because the Kingfisher knows how to move from one medium, air, into another medium, water, without making a splash. It knows how to move through that effortlessly and perfectly, and I'm going to show you that in a minute. So there's your European Kingfisher, and this is our North American Kingfisher. And you see they have very similar bills. Both of them have the habit of fluttering above the water until they see the fish that they want, right? And then they will plunge suddenly down, bill first. And when they do that, they make no splash. They make a splash when the whole body comes through, but then it's too late by that point for the fish, right? But the actual entrance of one medium into another is seamless. So the Japanese said, let's do it. So they put a Kingfisher bill on the Shinkansen 500. These things go over 200 miles an hour. Okay, I've been on one. And there we have it, Mount Fuji in the back. And there is a successful fishing expedition, no less than 3 fish on one jump. Right? And this one was also successful. We could talk about echolocation. This is an amazing way in which bats and whales, and especially the dolphins we're familiar with, use sonar or radar to move their way through either darkness or murky water. Who needs to see when you've got a perfect radar or echolocation system? And so the bats do it, the dolphins do it. We figured out how to do it. Man, mimicking God, realizing the brilliance of a system that can't possibly be improved on. Why do those airplanes have those little things on the end of their wings, winglets? Because we realize the birds do. And there's a reason for it. Notice this eagle with its winglets. If you look at the aerodynamics, a flat wing produces drag, but the up tip of the wing causes this to be greatly reduced. And the distance on a long-haul flight can be as much as 3% savings in fuel. Pretty cool. This vortex that happens on the straight wings is pretty much ablated by the winglet. And my second to the last slide, as I'm slightly over, that's a pretty good example. A peregrine falcon, I told you, can do 270 miles an hour in a stoop. That's straight down accelerating. They're accipiters, right? Which means that they're raptors that prey on other birds. And some of those other birds are very fast. Like a dove is very fast. So an accipiter, which is different from an eagle or, say, a hawk, has got to be able to intercept a moving object. So when you do that, you go 270 miles an hour, you can get the job done, all right? So the B-2 bomber on the bottom is built like that. And here's my final slide. O Lord, how manifold are thy works, in wisdom thou hast made them all. The earth is full of thy riches. Thank you for your patience. I'm sure that there was a lot to listen to. It felt like information overload right from the get-go. Again, my purpose wasn't to bowl you over, and I don't have all the answers, but it was to give you a sense that if you look into this, the more you look, the more convinced you are. It is rather daunting to be in countries where the educational system is so set against this and denies it and ridicules it. The first response you get when you attempt to engage them is not, well, we wish that could be true. We wish we could have a God to believe in. We just don't see the evidence for it. So regretfully, we have to live a meaningless, purposeless life. That's not what they say. The first thing you get is anger. You talk about the weather, no anger. Talk about sports, possible anger. Talk about politics, definitely possible anger. Talk about creation, livid anger. They don't want that God to exist. Now, can you be rational when you're angry? I ask that question. Let us then, shall I close this session in prayer, and then we'll have our break and try to tackle some questions after that.